Today we're going to do cell counts, and we're going to do a cell count using a hemocytometer and trypan blue. And then we're going to do the count under the microscope. Today I decided to do a 1 to 10 dilution for this cell count, and meaning that we're going to use 90 microliters of the trypan blue and 10 microliters of the cells. So in order to do that, we set up our pipette to 90 microliters, and we get our trypan blue ready, and we're going to add it at the bottom of the centrifuge. Right? We set our pipette to 10 microliters, and we have our cells ready. You're gonna first mix so that the cells are evenly distributed, and then you obtain the 10 microliters of cells. And you want to mix it into the trypan blue. You're gonna pipette up and down, and when you're ready, you're gonna get 10 microliters of your mixture, and we're gonna inject it into the hemocytometer. For this part, you wanna use your hand so you can have a steady hole into the micropipetter, and then you wanna go ahead and inject into the slide. This graph shows a representation of how the hemocytometer looks like under the microscope. This technique you will be using during cell count. And as you can see, we have 4 distinct quadrants right here. And in each quadrant you will find 16 other squares. When I'm doing cell counts, sometimes you encounter cells that are actually in between the lines, like shown on the figure. What you want to do is take 2 sides of your square, and you will count the cells that are there. But the other side, they won't count on your cell count. In this case, we have 1, 2, and 3 cells on our upper left corner. You will do the same to every other quadrant on your hemocytometer. So now that we have our samples in the microscope, we're going to do the cell count, and you want to locate 4 quadrants. Each quadrant will have in 16 squares, and you're going to count the cells in there. You're going to look around circles and really bright. Those are going to be your cells. However, you're also going to see cells that have the trypan blue injected inside. That means that you don't count those cells because the cells are dead. When you're ready to do your cell count, you're going to prepare your notebook in this way. You're going to have the concentration or the type of treatment that you're doing. In this case, I'm doing 50 picomolar BPA. I'm gonna do the cell count of this, and again I did a 1 to 10 dilution, 10 microliters of cells plus 90 microliters of trypan blue. When I'm reading the quadrant, I want to draw this cross to represent the 4 quadrants that I'm gonna be counting on. So when looking under the microscope, I have 5 cells on the upper left, 9 cells on the upper right, 1 cell on the lower right quadrant, and lastly 4 cells on the lower left. I'm gonna sum them up, give me 19 cells, and I'm gonna take the average divided by 4. This will give me 4.75. Now I'm gonna convert this by multiplying by 10 to the 4th. This will always be constant, meaning that I'm gonna convert my volume from microliters to mLs, and I'm gonna multiply by 10 because of my dilution factor that I did. This will give me an equal of 475,000 cells per ml.